Hello everyone, I'm by the names of Immunology, a student at Antisera Hungary, level 3, biomedical science. I'm going to talk about the types of Antisera produced and their application. And Antiseras are blood serum preparation that contains antibodies specific to a particular antigen. And the types of Antisera we have the polyclonal Antisera. It is generated by immunizing an animal with an antigen. The animal's immune system produces a range of antibodies that target different epitopes of the antigen. Secondly, we have monoclonal Antisera. These monoclonal Antisera are produced by the cloning a single B cell that is specific to a single epitope of an antigen. Now we go to the application of Antisera in immunoassays. We have the enzyme-linked immunoabsorbance assay, which is the ELISA. The application is used to detect and quantify antigens like e.g. the proteins, viruses, and hormones in a single sample. And Western blotting, the application is used to detect specific proteins in a sample after electrophoresis. The proteins are transferred to a membrane and antibodies are used to bind to the target protein, allowing its identification and quantification. Then the third one, we have radioimmunoassay. The radioimmunoassays are used to measure the concentration of antigens by detecting the amount of radioactivity in a sample. Then immunohistochemistry. This immunohistochemistry is used to visualize the location and quantity of specific antigens in sample in a tissue section. The antibodies are conjugated to enzymes or fluorescence tags that generate a visible signal when they bind to their target antigens in the tissue. Example, like often used in cancer diagnosis to detect the expression of tumor markers in biopsy samples. Then we go to immunoprecipitation. Antisera are used to isolate a target antigen from a sample by forming an antibody to antigen complex, which can be precipitated out of the solution. This technique is used for studying protein interaction and purification of antigens. Example can be used to purify specific proteins from cell lysates for further analysis. 6 will be immunofluorescence. The application Antisera can be used in immunofluorescence assays to detect the presence of antigens in cell or tissues. The antibodies are conjugated with fluorescent dyes, which emit fluorescence when excited by light. Then lateral flow immunoassays. Antiseras are used in lateral flow immunoassays such as pregnancy tests or rapid diagnostic tests for infections. The antibodies in the test strips capture and bind to the antigen of interest, providing a visible or visual signal. Then flow cytometry. Antisera are used in flow cytometry to analyze the characteristics of cells in a fluid stream. Antibodies conjugated to fluorescent dyes are used to detect specific antigens on the surface of cells, enabling their identification and quantification. Like for example, the flow cytometry is used in immunophenotyping of blood cells, such as in leukemia or lymphoma diagnosis. Then we go to the second question, which is the significance of monoclonal antibodies in immunoassay. Monoclonal antibodies play a significant role in immunoassay due to their high specificity or consistency. Like the first one, we have the high specificity. Monoclonal antibodies bind to only one specific epitope, reducing the likelihood of cross-reactivity with other molecules, which enhance the assay's accuracy. Then the second is the consistency. Unlike polyclonal antibodies, which can vary between batches, monoclonal antibodies are identical, ensuring reliable and reproducible results across different tests and times. Quantifications. Monoclonal antibodies for precise measurements of antigens levels in complex samples such as blood, serum, or tissue without interference from other components. Then we have the broad applications. Monoclonal antibodies are widely used in various immunoassays like ELISA, Western blotting, flow cytometry, and lateral flow assays, contributing to fields like diagnostic, cancer therapy, and research. Then we go to the third question, which says the immunoprecipitation, how it works and its application. Immunoprecipitation is a laboratory technique used to isolate and purify a specific antigen from a complex mixture by using antibodies that specifically bind to to that antigen. Then how it works, we have the sample preparation. Biological sample, whether such as cell lysis, serum, or tissue extraction, is prepared by breaking down the cells to release proteins. Then second step, we'll be having the antibody binding. A specific antibody that recognizes the target antigen is added to the sample. The antibody binds to its corresponding antigen, forming an antibody to antigen complex. Then binding to beads to facilitate the isolation of the complex. The antibodies usually linked to protein A, protein G, or protein L beads. Then incubation. The sample is incubated typically for 1 to 2 hours or overnight at 4 degrees Celsius to allow the antigen complex to form and ensure efficient binding. Then washing, which is the fifth step. After the antibody antigen complex are captured by the beads, the mixture is washed multiple times with a buffer to remove unbound proteins and other contaminants. Then elution. The antibody antigen complex is then eluted from the beads, usually by altering the conditions. Then the analysis. The target protein is then analyzed by gel electrophoresis, Western blotting, or other methods to identify the presence of the antigen or to assess its interference with other proteins. Then the applications of immunoprecipitation of the protein identification can be used to purify or isolate a specific protein. Then we have protein-to-protein interaction studies. Immunoprecipitation is commonly used in the study of protein-to-protein interaction by pulling down the protein with an antibody. Then targeted protein purification. It's often used to purify proteins from crude extractions or lysis. It is particularly used for isolating protein complexes that are difficult to purify through traditional chromatography methods. Then epitope tagging. In cases where a specific protein is tagged with an epitope, such as the flag HA or NYC tags, can be used to purify the tagged protein from a sample. Detection post-translational modifications. Immunoprecipitation can be used to study post-translational modifications of proteins such as acetylation or glycosylation. Characteristics protein complex can help to determine the composition of multiple protein complexes. Detection of pathogen proteins. Immunoprecipitation can be used to detect viral or bacterial proteins from infected cells or tissues. This is helpful for studying infectious disease, and understanding how pathogens interact with host cells machinery. Detection antibody-antigen complex. It's often used in immunology to identify and analyze antibody-antigen interactions. Validation of antigen specificity. When new antibodies are developed, immunoprecipitation can be used to confirm their specificity. If the antibody binds only to the target antigen, it can be considered validated for use in further application. The fifth question, which is the immunohistochemistry and how it's performed. Immunohistochemistry is a laboratory technique used to detect and visualize specific antigens within individual cells by using antibodies that bind specifically to the antigen of interest. The principle of immunohistochemistry relies on the specificity of the body of antibody-antigen interaction. Then the process we have is antigen-antibody binding, a primary antibody specific for the antigen. Then labeling, the primary antibody is conjugated with a detectable label to visualize the binding event. Then how immunohistochemistry is performed. We have cell culture and fixation. Cell cultures. Cells of interest are cultured on suitable substrates, then e.g. glass cover slides or multiple well plates. In fixation, cells can be fixed to preserve their structure and ensure the antigens of interest are stable and accessible. Then blocking agents such as bovine serum, albumin, or normal serum are applied to the cells to block non-specific binding sites. This prevents the antibodies from binding to components other than the target antigen and reduce background staining. Primary antibody incubation. The primary antibody, which specifically recognizes the target antigen or protein, is applied to the cells. This antibody can be either monoclonal or polyclonal. Then washing. After primary antibody incubation, the cells are washed several times with a buffer to remove unbound antibody and reduce background noise. Secondary antibody incubation. Secondary antibody incubation. If the primary antibody is not directly conjugated to a detectable level, a secondary antibody that recognizes the species of the primary antibody is used. The secondary antibody is conjugated to a detectable marker. And the washing again, the cells are washed thoroughly again to remove unbound secondary antibodies and prevent non-specific background staining. Visualization. For fluorescent-based detection, the cells are viewed under a fluorescence microscope when illuminated with a specific wavelength of light. The fluorescence marker attached to the secondary antibody will emit light at a different wavelength, making the antigen visible. Then we have the analysis and imaging. The final result is analyzed using a microscope. If fluorescence is used, the sample is observed under a fluorescence microscope, while enzyme-based ICC can be viewed under a light microscope. Images are typically captured using photomicrographs to document the location and intensity of the antigens of interest in the cells. And thanks.